You can start, Michael.
Hello, everybody in Brussels. Can you hear me? Am I audible?
We can hear you.
Wonderful. Thank you for the invitation to speak at this conference, especially allowing me to deliver this talk by an unconventional method. First of all, I would like to point out that my beginning slide has a very embarrassing error. According to my slide, I'm going to give you an Immortalist strategy, but actually I'm not Casanova. I actually am not good at providing people with a guide to immorality. That should read a new Immortalist strategy, not Immoralist strategy. So with that out of the way, let me begin. All right, those of you who know a little bit about my career will probably associate me with the work I did from 1976 to 1994, which was on the evolution of aging, the postponement of aging, and the physiology of postponed aging, all with fruit flies. Although the fruit fly, Drosophila, was used in order to provide a simple model system for testing general ideas which should apply to the evolution of aging in any species. Some of you may not know that since the mid-1990s, I changed my research focus from aging to the problem of biological immortality, in theory at first, and then experimentally in my lab, we have worked on changing the timing of the onset of biological immortality. And very recently, we have turned to the physiology of biological immortality. Again, all with Drosophila, the fruit fly. Now, along with my scientific research on the problems of the evolution of aging, since 1984, at the insistence of other people, I have been working on applications of my work to interventions with human aging, specifically, of course, to make human aging slower or better than it is normally. And my work on that problem, which may be of some interest to those of you in the audience today, has had three phases. The first phase, from 1984 to 1999, I call the evolutionary route to the Methuselah mouse. My idea in 1984, which I published in New Scientist, was to use evolutionary techniques to create a much longer-lived mouse, and then to use close physiological study of that longer-lived mouse as a model system in order to find ways to biochemically or otherwise medically intervene in the human aging process. And during this period of my work, about 15 years, I thought of this in much the same way as Aubrey de Grey, as a long-term plan taking 10 to 30 years from the start of research to the development of interventions that people could actually use. Those of you who have read my book, The Long Tomorrow, will know that in 1999, I realized that there was an alternative route to making Methuselah humans, which was the evolutionary genomic route, starting from the Methuselah flies I'd already created by laboratory selection, which I will describe shortly, and using genomic tools to connect the specific changes in the fruit fly to corresponding biochemical pathways in humans. In this second strategy, it is not assumed that things that work in Drosophila will also work in humans. The plan is instead to check whether or not any particular gene enzyme pathway is involved in both the fruit flies and the human Methuselahs. However, in the course of preparing a book for publication with my colleagues Larry Muller and Cassandra Rouser, I have developed a third strategy. Now, all three of these strategies, I think, are useful strategies. The distinctive part of my third strategy, which I call natural immortality, is that you can start on this particular type of intervention within 1 to 3 hours from the start of this talk. and as such, I think it is, in a sense, the best way to go to begin with. A simple way to view these three different strategies is that they will be available for use in reverse order. That what I'm going to talk about today is something you can start tonight. The plan I've had since 1999 is something that could yield results in a few years, but ultimately we do in fact want a Methuselah mouse created by evolution for refining what we're doing with the first two plans. Today I will be giving the scientific background for this third plan for intervening in the human aging process and proceeding toward indefinite human lifespans. Here is my outline for my talk. I will begin by reviewing some basic principles as to how evolution by natural selection can give some species biological immortality, and other species unfortunately suffer from aging, species like ourselves, which suffer from a falling force of natural selection. Despite that, what I've been working on since the mid-90s is the phenomenon of biological immortality as it arises at late ages. This is now well documented in humans over the age of 90. Interestingly, in laboratory insect species, biological immortality arises much earlier in the cohorts proceeding through biological time as adults. And as I hope to show you today, we can understand this phenomenon of later life biological immortality, again using evolution by natural selection. In the third part of my talk, I will discuss the recent research we published primarily in Nature last month on the genomic machinery that underlies the evolution of aging and biological immortality. This is the part of the talk that some people might find depressing or challenging, and I will be happy to answer questions about that at the end. And at the conclusion of my presentation, which I'm sorry will be somewhat long, that is where I will provide details on the new strategy, the natural immortality strategy that I have developed for starting people in the direction of indefinite lifespans. And I will wake up those of you who have fallen asleep at that point, perhaps with a loud bang. So I'll begin essentially with a review of work for which I'm fairly well known, I think is fairly well understood. This is for the sake of those of you who have not yet been exposed to my research in the 70s, 80s. The first thing to understand, and this is a very important point to understand, is that biological immortality is something that happens in nature in the same way that fusion happens in nature in the sun. And that's going to be my starting point. It's very important to understand that there is nothing in the molecular or cell biology of eukaryotic organisms that prevents indefinite lifespans or biological immortality. And here we have a dramatic example, a particular bush shown on the left in this photograph. It's a kind of interconnected root system. The round or roundish, squarish structure in the surface is the above ground part of this bush. This bush has been dated using radioisotope methods as having been there growing for more than 10,000 years. It grows in the Mojave Desert of California, which is the desert you drive through when you're going from Los Angeles to Las Vegas. The fact that this bush can be sitting there for 10,000 years in reasonable health and continuing to grow, I think shows you some of the potential of life to proceed without aging, which is the definition of biological immortality that I use. In this next slide, I show another type of organism that can live indefinitely, which is a fissile sea anemone, a rather pretty organism. Basically, the simple explanation is to say that if there is no difference between the products of fissile division in the course of reproduction and there is no sexual reproduction, then natural selection favors immortality. And there's a simple way to understand this, and that is that if you have reproduction strictly by fission or splitting in two, If aging were to occur in the products of symmetrical fission, that would imply that all individuals in the population and all their descendants would progressively die off, which would lead to the extinction of the species. So in other words, in this situation, evolution by natural selection will eliminate life forms that undergo aging in all of the symmetrical products of fission. And this can occur in plants, in multicellular animals, and of course in unicellular organisms as well. So there's really nothing about molecular cell biology that requires aging in any sense. Just as fusion is a byproduct of E equals mc squared, and you can see it in the sky when it's a sunny day, in the same way in these organisms, evolution by natural selection can produce unlimited lifespans. Therefore, there's no absolute molecular cell biological reason why humans could not have absolute lifespans, at least in principle. Now, of course, we do not have such indefinite lifespans. And here in this slide, I'm showing, most importantly, the death rate per 100,000 individuals at any one age due to all causes in this purple color here. Cardiovascular disease in red, appropriately showing a very similar pattern of a marked increase with age in the incidence of death due to that particular cause. Cancer less dramatic, on this scale at least. So what this is showing basically is a simplified sketch of the physiology of aging, and in particular how it kills us, which is the most dire aspect of aging. Here is how people like myself have been explaining the evolution of aging since 1966 and the work of William Hamilton. What Hamilton showed in 1966 was the intensity of natural selection acting on age-specific, proportionally uniform changes in your survival probability qualitatively looks like this. The force of natural selection is at 100%, is very powerful before the first stage of reproduction, little b, in a population. In a population, which has its reproduction starting at age b, from then until the cessation of all reproduction in the population, the force of natural selection steadily falls with biological age, until finally it is flat at 0%, and it stays at that value from little d until infinity. Now that is conceptually how Hamilton explained aging, although he didn't completely work out the mathematics, and he didn't supply any direct experimental evidence for this. My advisor, Brian Charlesworth, in the 1970s, showed that mathematically, using explicit evolutionary genetic theory, that this does in fact have the effects that Hamilton suggested qualitatively it would. That is to say, in species that have this pattern of the force of natural selection, sometime after the age a little b, physiology and survival are expected to deteriorate with age. Now, because this is a Teleplace talk, I cannot show you the following animation, but what we have instead here is the concluding frame of the animation. Crudely speaking, what this shows is that before the period of reproduction in a population, if you have a gene that's going to kill you with 100% probability, it will kill itself off in the population. It will not be passed on to subsequent generations. Conversely, if you have a similar kind of gene, but it kills you after the cessation of reproduction, in terms of the population's pattern of reproduction, then evolution by natural selection is entirely indifferent to you dying off of that lethal gene. And it's in this reproductive phase where you get the quantitative reduction in your survival probability. Now, a basic corollary of this type of theory is the evolution of aging in all strictly non-fissile organisms. So for example, this would include all mammals and all insects. All organisms that reproduce by a small single-celled stage or equivalent, which we can call an egg for our purposes today, focus on animals. Whenever you have a cycle like this where the immature organism grows up, becomes an adult, and the adult reproduces by producing very small numbers of cells, normally just a single cell, from which the life cycle starts again. And that adult does not ever split in two. That adult will progressively deteriorate even under ideal conditions. That is the prediction of this theory. What I realized in the late 1970s was that if I shifted the time of reproduction, which means moving this particular box from the left to the right like this, the pre-reproductive period in which natural selection is most intense would clean up the deleterious alleles or genes or mutations that have bad effects early in life and produce a longer, more robust lifespan. So that was my theoretical expectation. And being an experimentalist, what I then wanted to do was to test if this prediction was true. And in an experiment that's now relatively famous, I showed that it was true first in the 1970s. And I used my lovable fruit flies shown here. That's the adult form of the fruit fly, which most of you know. The fruit fly produces a small white egg, which develops into a tiny larva or maggot, which then pupates to produce the adult. In a laboratory setting, what we normally do is we take the young adults at 2 weeks of age, which will have mated by then. We let them lay eggs in a new culture vessel, usually a vial or a population cage, and then we remove the adults after they've laid all their eggs and let the juvenile cohort grow up to reach adulthood again. And we repeat this generation after generation after generation. In this case, this age at which reproduction starts is very low. So in populations like this of fruit fly, the prediction is that aging should start early and proceed fairly quickly. By contrast, in my laboratory starting in the 1970s, what we've done is this kind of experiment in which we discard the eggs laid by young and even middle-aged adults and only let the oldest adults reproduce. That's shown here. So only at this later stage in adult life are eggs produced that will start the next generation. In quantitative terms, what we've done is we moved little b, the first stage of reproduction in the population, to much later ages, and our expectation was that after enough generations of natural selection, aging should be slowed or postponed. So what we have here is a contrast between early reproduction, in which we expect fast aging, and delayed reproduction over many generations, in which we expect the evolution of slowed aging. And these are known as selection regimes B and O. And I may occasionally use those terms. About 80 generations into one of these experiments, here you have the pattern of survival in the males that are bred normally with with early reproduction. And here you have similar data for females. And then in the green line in the upper plot and the mauve or dark blue line in the lower plot, we have male and female reproductive patterns after 80 generations of being reproduced with, at later ages, delayed reproduction. So this corroborated the prediction that I started out with and supports evolutionary theory. Now, a non-trivial aspect of our work in the first 15 years or so that we were doing this kind of research is that there are, in addition to increased rates of survival to later ages, there is also a number of physiological enhancements in longer-lived flies, which we spent much of the 80s and 90s studying. These longer-lived flies have greater fecundity and fertility later in adult life. Males are better at mating later in life. These flies also have better resistance to acute stresses like alcohol, desiccation, starvation. They have improved functional performance, better flight duration, and later locomotion. They do, however, have some reductions in performance, like reduced early fecundity, specifically under bad conditions and slightly slower development. This will prove to be important later basically, as I will show you, you cannot disconnect aging from development or indeed the later phase of biological immortality. But the meaning of this result is that this experiment, which has since been done a number of times in a variety of laboratories, has shown that Hamilton's basic theory of the declining force of natural selection as the explanation of aging was the correct theory of aging, and still is. And an obvious demonstration of the validity of this theory is that we can take this theory and immediately proceed to experiments that manipulate aging in any organism that is experimentally tractable. And it is notable that this is different from most other theories of aging, which do not provide such easy work for experimentalists. Now that was all by way of review for those of you who have not yet been exposed to my work. What I'd like to show you now is the work we've done over the last 15 years, of course, which I'm more excited about, and that is work on the phenomenon which we call sometimes later life biological immortality, when we're not upsetting many conventional biologists who don't believe in immortality. Remember that when I use the term biological immortality, I simply mean the absence of aging, the absence of a tendency to die off at greater and greater rates as a function of age. If there were no biological immortality and aging proceeded unabated as a function of adult age, then this delightful French woman, Madame Jeanne Calment, who lived to 122 years, would not in fact have lived past 100 years. So the real scientific question is, how was her feat possible? How is it possible for people to live so long? And basically, here's the answer. This is the age-specific death rate of English women in 1900s as a function of their age. And basically, these data show that starting sometime in the fairly early 90s, in terms of their years of chronological age, the mortality rates are essentially flat. They become more variable with age only because of the statistical effect of the law of large numbers. There are more people with which we can estimate the average mortality rate at earlier ages in this phase compared to later ages. In effect, these women are exponentially dying off, indicating a stable mortality rate, very different from the pattern from the ages of 25 to 85, which I showed you earlier, in which rates of death go up exponentially. And this will appear in our forthcoming book, Does Aging Stop? So basically, if you were to extrapolate from our rates of death from aging, which rise from 25 to 85 years of age, there would be virtually no chance of a human being living to 110 years of age, much less 122. Now, humans are a very problematic organism for the experimentalist. It's very hard to keep thousands of them in a laboratory cage, particularly from a funding standpoint. So, of course, the best experiments come with laboratory animals. And 1992 marks the year when basically many of us, such as myself, who did not really believe at that time in the idea of later life biological immortality, this was the year in which we were woken up by two publications that appeared in the journal Science. And this was in laboratory cohorts of fruit flies and medflies. Here's the fruit fly, Drosophila melanogaster. And here's their age-specific mortality pattern as a function of adult age in days. And here you see this is a logarithmic scale, which means that a roughly straight line here corresponds to an exponential increase in death rates, which comes to an end in this particular cohort at around 35 days of adult age, and then it fluctuates radically as the last of these flies continue in life. Even more staggering experiment was done by Jim Carey, University of California, Davis, where some of the cohorts that he worked with, for example, here in experiment 3 in the bottom, involved starting his cohorts with hundreds of thousands of medflies in gigantic walk-in cages. And here you see the roughly exponential increase in mortality rates corresponding to the aging phase, and then you see a protracted and very stable pattern of survival continuing for quite a number of days of adult age And again, the data break down at later ages due to fewer and fewer flies being alive and less accuracy of estimation of mortality rates. So when I first saw these data, I was completely flummoxed by them. I could not believe these results. And basically, I puzzled over that for about 2 years until I developed a crude idea. And the thing to understand is that this is what's called the Gompertz equation for the exponentially accelerating rate of death as a function of age, where E is the natural exponential function. Capital A is the normal death rate at the start of the adult phase, and alpha, shown here, gives you the exponential acceleration coefficient in the rate of aging. Basically, what my colleagues Jim Carey and Jim Kurtzinger showed was that this equation breaks down. So why does it break down? So crudely speaking, we can divide life into 3 phases: development, before the onset of reproduction of population; aging, the first phase of adulthood; and then what I call an immortal phase, in which mortality measures stabilize. Doesn't really matter what scale you use, logarithmic or linear, your mortality measures will crudely stabilize. This does not mean perfect flatness. It means, however, the absence of the persistent, sustained, generally exponential rise in death rates associated with the aging phase. That is, in effect, the problem that had to be solved. And here was my first intuition, and that is that if you recall the form of Hamilton's force of natural selection equation, after the last age of reproduction in a population, it is flat forever. So I was wondering, from thinking about this equation, whether or not it would be true that aging is a result of, in fact, a decline in Hamilton's force of natural selection, and the late life stabilization of death rates was due to the stabilization in the fall of Hamilton's force of natural selection. A simple way of thinking of this is if you invert the force of natural selection and instead plot it as the weakness of natural selection. Early in life, there is low weakness of natural selection. The weakness then increases during the aging phase, the reproductive phase, and some period afterward. And then after the end of reproduction, the weakness of natural selection stabilizes. So if this is basically the cause of both aging and its cessation, then if we simulate formally the action of natural selection over a lifespan that includes a post-reproductive phase, then what we should see is biological mortality. Now, my colleague Larry Muller did these simulations, and here is the result of throwing a whole bunch of genetic variation at a theoretical evolving population with an early adult phase here of very low mortality rates, on first an exponentially increasing mortality rate, it linearizes, and then it stabilizes in late life. And in fact, this type of result from simulating the consequences of Hamilton's theory explicitly at all adult ages shows the deceleration or the virtual cessation of aging at late life. We published this theoretical result in 1996 in the Proceedings of the National Academy of Sciences of the United States. But of course, because I'm an experimentalist, I immediately wanted to test this theory and see if I could use an experimental technique or trick to manipulate the ages at which there is a transition from aging to biological immortality, as I've defined it here. And this is what we show here. This is a result published in 2002 in the journal Evolution. This is the CO population. This is female data, cohorts of aging females, 5 different cohorts of this type. This is a logarithmic scale, the natural log. Here you have the aging phase, the pronounced exponential rises in death rates, then it slows and basically finally plateaus at stable death rates. Then what we did from these populations was we derived populations that a much earlier cessation of reproduction. These are the gold colored cohorts here. Aging was faster, and they reach the immortal phase earlier, which is what the type of theory I showed you earlier requires. So this was what we call in science a critical test or an attempted falsification of the theory of biological immortality that I just showed you. And in fact, what this shows is that experimentally, we can tune the age at which the biological immortality starts by shifting the last age of reproduction in a population to an earlier point. Now bear this in mind because this will turn out to be significant for human evolution, which I will be coming to shortly. So what we have shown is that biological immortality evolves as defined by Hamilton's forces of natural selection. In particular, the aging stops the level of individuals, a phenomenon we can explain evolutionarily, and a hypothesis we have tested experimentally. Not only does biological immortality exist, that is to say aging does stop, but the age at which this transition occurs can be made to evolve experimentally. This shows that in terms of the absolute foundations of biology, not only do some species not age, but even species that age go from an aging phase to a period of biological immortality, and the age of that transition is not fixed by evolution. So that basic concept I thought was incredibly exciting from an immortality standpoint. Now, one of the things I'm often asked about is what I've shown here is what happens at the level of macro theory and macro experiments. That is to say, at the level of evolutionary theory and experimental evolution, we can easily explain the evolution of aging and mortality, and we can evolutionarily manipulate the pattern of aging and immortality. So you might be saying, well, great, let's find the 3 or 4 genes that, or pathways that determine the process of aging that stop when immortality arises and stop those processes of aging early. And what I'm now going to show you is why that in fact won't work. And basically this is because the foundations of these evolutionary patterns is a very complex world of microcausation, or in other words, the underlying genomic substratum. Now, before the advent of genomics around the year 2000, meaning whole genome studies of genetic variation, the genetics of aging, like the genetics of most other characters, I would compare to using a flashlight in a dark factory. In a dark factory, if you have a flashlight and you're wandering around the factory, when you focus the flashlight in on a single piece of machinery, you can study that machinery in detail and learn a great deal about that particular piece of machinery, which is what geneticists love to do. So some of this machinery, for example, in the context of aging, involves free radical damage, and there's a great deal of quite good experimental evidence for the role of free radical damage in aging. But you're just looking at that one piece of the metabolic machinery. You can add up some of these specific pieces of machinery and come up with ideas like the 7 deadly sins of sins. But unfortunately, what I'm going to show you is what happens when you turn the floodlights on in the factory and you see all the machinery of aging. Now, we've been studying this problem for a long time, actually about 20 years. And our first experimentation involved things really now old-fashioned technologies like protein electrophoresis and the identification and characterization of single nucleotide polymorphisms, or SNPs. And our last paper of this type was published just in 2009 in Nature Genetics. One of the exciting but also problematic aspects of this research is that it is easy to find loci, genetic loci, that are differentiated for enzymes involved in aging, enzymes that affect things like energetic metabolism and other well-defined physiological pathways controlling aging. In a sense, it's extremely easy. So what we needed to do was take a genome-wide look at what happens in experimental evolution of patterns of aging. And we did this using the ACO and CO populations I showed you earlier, the blue and gold differentiated populations, where the ACO populations had evolved for 600 generations to produce the radically different pattern of aging earlier onset of biological mortality. My colleagues at UCI, particularly Tony Long and Kevin Thornton, and my graduate student Molly Burke, analyzed whole genome data across all the euchromatin of the Drosophila involved in this experimental evolution project. So this is just to remind you of what we've got. We're comparing the genomics of these populations, the CO populations, with the genomics of these populations, the ACO populations, which at the time of this experiment, the ACO populations had about half of the lifespan of the CO populations, thanks to experimental evolution. And here is a genome-wide scan, which we published in Nature last month, where this dotted line is a very conservative false discovery rate threshold for significance. And whenever this line spikes above this dotted line, the solid black line spikes above, you have a region of the genome which is involved in the transformation of the life history of the ACO populations, producing their faster development, faster aging, and earlier onset of biological mortality. And crudely speaking, what you can see here is many, many spikes distributed widely over the genome. I'll skip this slide. The main significance of this slide is at the bottom. Just at the level of structural genetic variation, we have found non-synonymous single nucleotide polymorphism frequency changes at 506 genes. What this means is that more than 500 genes change in their frequencies in response to the experimental evolution of development, aging, and biological immortality in these populations. 575 of these genes have known gene ontology terms and are actually pretty well characterized, at least in Drosophila. So here's your problem, assuming you want to do something about your biological aging. If biological aging were due to a few simple and well-defined pathways, like this serotonin synthesis pathway shown here, then intervening in this pathway to speed it up or slow it down by adding supplements or genetically modifying perhaps the enzymes involved in this pathway would be a reasonable project. What the genomic results I'm just showing you indicate that the project is instead at least this complicated in that it depends on complex genomic networks. In fact, a fair characterization is probably this, taking into account structural and regulatory genetic variation, that the genomics that underlie aging is this physiologically complex, where this is a gigantic genomic network diagram, not from Drosophila. So what to do about all of this? Here's where I come to the payoff, and let me just say to people, those of you who've been sleeping, wake up, wake up. It's time for you to pay attention if you've been sleeping through this evolutionary biology. Here's the idea. This idea essentially was pretty clear to me and my colleagues around 10 years ago. The idea that the single best thing you could do with aging is to stop it. And the single best way you could do that would be by shifting the onset of the immortal phase to an earlier point in chronological age. So you stop the increases in mortality and physiological dysfunction that are associated with aging. And once you stop aging earlier, you then try to compress the mortality associated with aging so that the steep incline becomes more and more shallow, and you would slowly converge on biological immortality starting at a much younger age. Now that is the sort of technological fantasy, if you will. When I thought of this crude idea in around the year 2000, I had precisely no idea as to how this might be done without using experimental evolution, of course, because at no point in my career have I proposed that we make human beings evolve differently than we do normally. Now, let me just show you one example of what the physiology of late life or biological immortality looks like. This is work that is now under review from my graduate student, Parveen Shahrastani. Now, what she showed was for a functional character like the amount of time you can move around, of course, when you're a fruit fly, is that during the aging phase, you become progressively more and more immobile, and then we skip this intervening phase for statistical reasons. Once you're firmly on the later life plateau in mortality, what we call biological immortality, your physiological decline has stopped. And there may even be a slight enhancement of this particular function in individuals alive during late life. This is not true of all physiological characters. Some continue to decline during late life. Some, interestingly enough, decline even faster. But what this shows is that not only in terms of mortality, but also in terms of at least some functions, during the immortal phase, your function stabilizes, at least in some respects. Now, one of the things we turned up sort of accidentally in our work with Drosophila is the timing of the cessation of aging is not only something that can be manipulated using experimental evolution, it is also something you can manipulate by changing the environment in which the fruit flies live. You can change the transition to biological mortality by changing the food regime and by changing lifestyle. In particular, if you're a female fruit fly, we can change when you hit the phase of biological immortality by changing your access to hot young males who are ready to mate with you, which in my lab we call the Joan Collins effect, but many of you are probably too young to remember who Joan Collins is. So this raises the question of whether or not there is a lifestyle for humans that could environmentally produce an earlier transition to biological immortality. And I at first couldn't think of what that might be at all. But here's the key concept, and the key concept is what is natural for humans? Well, for humans as for any other species, the natural is what we are best adapted to. And of course, adaptation is a product of the long-term action of natural selection. So in particular, what is unnatural and what we are not adapted to is what I will call the industrial lifestyle, where you get up in the morning, you walk a few feet to your car, you drive your car to work, you walk a few feet from your car to your workplace, you sit in front of your computer screen, as I'm doing now, you eat your Twinkies and other junk food, you drive back home using your car, when you get home you sit in front of the television eating more junk food, and then finally you go to sleep and the cycle repeats. Well, this is a novel lifestyle that is only about a century old, and we are not adapted to it. That's only about 4 generations, not enough selection to adapt us to things like automobiles and Twinkies and television. So there are only 2 significant candidates for what would be a natural lifestyle for us, which we would be well adapted to. One is the agricultural lifestyle, which Eurasian populations have had for about 10,000 years, and the other is the hunter-gatherer lifestyle, which there are still a few remaining populations of hundreds of individuals that still practice this lifestyle. Which of these 2 is natural? Well, we've actually worked on this problem in experimental evolution, particularly my colleague, Dr. Margarita Matos of the University of Lisbon, has shown that populations given a new environment adapt really well to that environment in 30 to 60 generations. Certainly the hundreds of generations of the agricultural diet, which Eurasians have had, should be enough to make us very well adapted to the agricultural lifestyles where many of our calories come from grains, where we have daily manual labor, and fairly high population densities compared to the ancestral hunter-gatherer tradition. For this reason, I was not a huge fan of the paleo diet, and this will be very significant now. Now, at this point, Julio promised me he would display on a screen in Teleplace the missing figure here, which is the all-important figure for this talk. Julio, will you display this figure in the conference hall, please? The basic idea of the figure that I can't see, but perhaps you can, is that young people of Eurasian ancestry are well adapted to agricultural lifestyles because at those early ages we had very intense natural selection. So a corollary of that inference is that people under the age of 30 can go ahead and eat grains and dairy products if that is your ancestry. Of course, if you are an Inuit from northern Canada or a hunter-gatherer in terms of your ancestry in sub-Saharan Africa, you should not eat agricultural foods because you do not have a history of being adapted to them.
Sorry,
But
we
the
can still
significant
not
point,
see. We can still not see the figure.
right, and I can't do anything about that, Sven. It's up to Giulio, who promised me he would do
Sven,
this for me at this point in my talk.
what you should do is to zoom on the last screen on your left that you're seeing now.
Giulio, is that something I do or Sven does?
No, it's something that only Sven can do. Something that only Sven can do
Okay,
because he has
Sven.
to zoom for the audience. That would be not the main screen, not the one immediately at the left, the one on the left, where on the vertical axis is written phenotypic value, and on the horizontal axis is written age. Yeah, that one.
Thank you. So while Sven is doing that, what I'm going to say is what this graph shows is that if you have undergone a relatively recent evolutionary change and you have a normal life history pattern of most reproduction occurring early in adulthood, then your youthful physiology will be adapted to the novel environment, but later in life, your older physiology will not be adapted to your novel environment. What this implies is that people my age, and I'm 55, are better adapted to the hunter-gatherer way of life than the agricultural way of life. 30 years ago, that was not true of me and like other Eurasians, but now it is true. And what that implies is the need to switch your lifestyle at later ages in order to preserve reasonable health. So Sven, have you succeeded in bringing up the slide for your audience?
Yeah, it's on.
Very good. So basically what you should be looking at is two solid lines, which are curved, and in between them a dashed line. And the dashed line is showing you that at earlier ages, the novel environment is what you're better adapted to. Then the dashed line switches back to the ancestral environment, the lower curve, and that is basically, that becomes your more natural lifestyle. Now, this is very exciting from the standpoint of evolutionary theory, because if you're on an agricultural lifestyle throughout your adult life, like most of us are, then the data show that we hit the immortality plateau very late in life in very bad health. But we have good evolutionary reasons for believing that hunter-gatherer populations, in fact, hit the late life plateau much earlier and in much better health. And there are three reasons for this. The first one is that hunter-gatherers have had a history of millions of years of adapting to that environment, at least 1 million years, possibly 2. Probably more than 2 million years ago, we were not hunting, but just gathering. This gives evolution the opportunity to do a very good job late in life, despite the great weakness of natural selection. In addition, what we know from a very strong theoretical result is the smaller your effective population size, you can think of that as a number of people you could possibly mate with, that you could meet in the course of a week. The earlier, that is to say, the smaller the effective population size, the earlier you plateau, the earlier you reach biological immortality. And it is certainly the case that hunter-gatherer populations had much smaller effective population sizes, which would have given them much earlier transitions to biological mortality. Finally, that type of environment features a great deal of what I will call accidental mortality, and that means most especially things like homicide, but also it includes things like death due to accidental trauma, death due to being eaten by an even bigger carnivore than us. Those are things that we have systematically reduced by adopting agricultural or civilized lifestyles. Basically, it's hard to reach a ripe old age in the Brazilian jungle, simply put. All of these things imply that if we make the transition to a hunter-gatherer diet later in life, over the age of 40, not only will we slow our rate of aging and improve our health, we will hit biological immortality earlier and in better health than we do now, shown up there, in Eurasian populations. This leads to the following recipe for what I call natural immortality. If you're over the age of 40 and Eurasian in ancestry, make a transition to a hunter-gatherer lifestyle. Now, there are many books about this right now. They're not entirely accurate from the standpoint of good evolutionary research, but they give you roughly the right idea, which means no cereals, no dairy products, at least moderate sustained activity levels, which you can call exercise, every day. Having done this, you will have enhanced your fundamental propensity to survive. You can then use all the tools modern medicine supplies us, unlike hunter-gatherer populations, to even further lower your mortality level during your last decades of aging and during the plateau. These are two things you can do right away. Then later on, when we can have things like autologous tissue repair from your own stem cells, you can repair the incidental accidents and damage that will inevitably occur. And by accidents and damage, I include things like ischemic cardiovascular events, such as minor heart attacks and strokes. I also include cancer. Nothing about these changes in your lifestyle will obliterate the possibility of cancer or cardiovascular disease episodes. They just make them less probable. You will still need to be repaired when those things happen to you. And finally, what this suggests we need is a new generation of pharmaceuticals with less side effects to continue this pattern of further depressing our mortality rates, given that they are going to be stabilizing at earlier ages. Together, all of these things supply the prospect of indefinite human lifespans. I would like to acknowledge support from a whole bunch of government agencies in multiple countries, both Canada and the United States. The demographic research, the macro level research, was done in conjunction with Larry Muller, Katsi Rauser, Ida Matos at the University of Lisbon, and my graduate student Parveen Shavastani. The genomic research done in collaboration with my former graduate student Enrique Teotonio, Joseph Denham at USC, Kevin Thornton, Tony Long, and most importantly, Molly Burke, all at UCI. Thank you very much for your attention. I hope that some of you are still awake.
Sven, you may want to relay some questions from the audience if you have scheduled some question and answer time, or you move directly to the next talk.
I'm sorry, Sven, if you are speaking, I cannot hear you.
Dutch. Ah, Michael, do you hear me now?
I can now hear you.
Okay, great. That means that I don't have to type on Sven's weird Dutch keyboard. As this end, the audio is only from a headset mic. We'll need to do questions by proxy. So I now open this to, yes, Danila? Okay, so firstly, what do you... actually, it's probably going to be easier if you just come in before I have to remember everybody's very long questions. So, yeah, if you have questions, ladies and gents, if you could possibly make your way towards the front of the auditorium, then you can field your questions directly.
Thank you very much for the presentation, Michael. The question is, first, do you support the deeper reasoning for evolution of aging as outlined, for example, by Holiday, that an organism has a limited amount of energy which it has to spread between reproduction and regeneration?
Yes, this is called the disposable soma theory by Tom Kirkwood. It has been discussed by evolutionary theorists since 1946, first by Peter Medawar, then particularly by George Williams. In around 1980, I defined this idea as antagonistic pleiotropy, which is the sort of population genetics generalization of the concept you have just brought forward. Over the course of my career, we have produced a great deal of evidence in support of this idea that trade-offs between different parts of life history and physiological function, especially trade-offs involving energy, are very important for the evolution of aging.
Okay, and then the second part of the question. In your presentation, you described a very fascinating phase in life, the late life, the biological mortality. The question is, have you tried to influence the parameters of the physiological parameters of the late life, influence them more directly, that is without shifting reproduction, but selecting just for the quality of the physiology during the late life?
No, we have not.
Why?
We have done experiments like that on aging. So we have selected on specific physiological characters that are important in the aging phase. And we have shown that selecting on those specific physiological characters does in fact change the aging phase. It would be, we are actually doing an experiment like this again, in which we will hope to track the impact of such physiologically focused selection on late life. This is the work of one of my new graduate students, Marta Santos.
Okay, thank you.
Hello. Hello.
Hello.
Yes, Eduardo Bonnet. We've seen each other. So I have a question concerning the concept or fact or in between that aging is an adaptation or lack of adaptation, and that therefore it's a mixture of many things, and that it's nonsense to hope to fight aging with a few simple strategies. As you know, your fruit flies after 80 generations live about twice as long as the initial strain. In labs, there has been other surprise life improvements. For example, in worms, almost 10 times life improvements just by knocking out one single gene. How are you able to match those two concepts together?
I have written several things about that specific issue. One of the best papers I was involved in was published in 2006 with my colleagues Wayne Van Voorhees and Jim Kurtzinger. If you look carefully at mutants that where you have single or double genetic changes, and they live much longer, you will very consistently find that they have impairments in reproduction, metabolism, competitive ability, or some other biological function. Results like this were actually first published by John Maynard Smith more than 50 years ago, where he showed that a variety of manipulations that reduce or eliminate reproduction are associated with very large increases in lifespan. This is a very long-standing result, and I think it illustrates the same principle that my first questioner referred to, which I call antagonistic pleiotropy, other people call trade-offs, and some other people call the disposable soma. It is very expensive to sustain the capacity to reproduce. It is expensive to sustain the capacity to compete. And if you reduce those capacities, there may be some benefits in terms of your survival. Now that, of course, is why a big part of why caloric restriction works, because in caloric restriction, you reduce your reproductive rate or capacity, and in so doing, you get some benefits for survival. So to me, there is actually no fundamental problem with the types of mutations, which is sometimes called longevity mutants, that you've brought forward. Such phenomena are pretty well understood, I think, genetically and mechanistically.
No more questions, Sven?
Thank you very much. Goodbye.
All right. Professor Rose, are you still with us?
Oh, there are questions. Oh.
There, there still are. There
Sorry.
seemed to have been a technical problem. I spent a lot of time thanking you and congratulating you for an excellent presentation. You won't hear any of this again. But the question related to the context in which this conference happens, this is an Immortality Institute conference, and we are concerned that the word immortality is a very challenging concept to use in a scientific context. Now, you seem to be comfortable with using it and describing proper science with it. Do you have any advice on whether the Immortality Institute should reconsider its name or should reconsider the way it uses the word?
Oh, well, I guess the one thing that's very important to always be clear about is that I'm assuming the Immortality Institute does not believe in the immortality of Greek gods who could be cut in two or burned or otherwise, messed with, and yet continue to survive. Any form of life can be destroyed given sufficiently extreme conditions. The only reasonable definition of biological immortality would be the absence of a sustained tendency to die off as a function of time. That is to say, biological immortality means stable survival rates with no aging, or very little. To me, the scientific question of immortality is a completely legitimate one, since you can see biologically immortal organisms if you go to the seaside and look at sea anemones, or you go to the Mojave Desert in California. Saying that is somehow not something we're allowed to discuss reminds me of the Middle Ages and the Catholic Church and the Inquisition persecuting Galileo for his belief in Copernicus's heliocentric theory of the solar system. The second point I would make is that there's also the technological question of human beings achieving indefinite lifespans. And I feel that the best way to do that is to achieve a state of biological immortality. Now, right now, we have people alive today who, in my terminology, have achieved biological immortality. Those are humans over the age of 95 or so. But they are in extremely poor condition and have very high death rates. So the technological challenge to me is finding a way for us to achieve biological immortality younger and in much better health so that we can sustain that type of health for an indefinite period.
Thank you. So, without wanting to put words in your mouth, you would advise us to not be afraid of using the word immortality, but to use it with caution.
Right. It's just like Darwin was very careful about his use of the word evolution because it offended so many people. Well, after 150 years of Darwin, we're very happy talking about evolution, most of us scientists anyway. So probably in 150 years, people will be very happy talking about biological immortality. It'll be no big deal.
This might be a good note to end on, but we do have one more question. Is that okay? Right.
Hello, Dr. Rose. My name is Erik Wolraves. I'm going to be honest, I didn't understand everything. Your presentation was probably the most complex we've heard yet. There's one thing I'd like to ask. Given the statistical death of aging rate of people past a certain age, 100 years and so, do you actually know, or do some people know why that is? Because reducing that age to a more interesting age, when we can still drive a bicycle and stuff, I mean, that's nice, but we don't know what happened, or do you?
I think what you are asking me is, what is the physiological mechanism behind the achievement of biological immortality? Am I correct?
If you know that answer, yes. I mean,
right.
I'm sure if you did know.
Yeah.
Okay. No, it's okay. Sorry.
Thank you very much for your question, because it actually brings to mind something I should have said earlier, and that is the following point. Aging involves physiology. The achievement of biological immortality involves physiology. But the causes of aging and the causes of the transition to biological immortality are no more physiological than the causes of adaptation. Adaptation, which is something evolutionary biologists have studied for the last 150 years, is the shaping of all of the attributes of the organism towards survival and reproduction. When we study aging and biological immortality, we are dealing with features of adaptation. In the course of aging, the detuning of that adaptation. In the case of biological immortality, what you might call residual or background adaptation. All of these things involve physiology, but are not merely caused by physiology. It's physiology shaped by the forces of natural selection. So there is no simple physiological why to aging any more than there is a physiological why to the transition from aging to biological immortality. That is what would be called in philosophy a category mistake. Just as before 1859, before Darwin's Origin of Species, virtually all biologists interpreted adaptation strictly in terms of physiological mechanism or God's intervention, one of the two. After 1859, we understood adaptation was a product of evolution by natural selection. Since 1966, evolutionary biologists following William Hamilton have understood that aging is a product also of evolution by natural selection.
Okay, thank you, Michael. On behalf of everybody here, I realized that because our communication to you is only via headset microphone, that you probably could not hear the rapturous applause at the end of your main part of your presentation, but I assure you it's been very much appreciated, especially you agreeing to come and deliver your presentation via this unusual means. So we're a little, I guess, getting on for half an hour over our kicking out time of the venue, but thank you very much for coming, and until the next.
Thank you very much for inviting me. Goodbye.